thank you for joining us today for our webinar, our Koolen-de Syndrome Foundation webinar. Normally we have Dr. Anna Falzer here, but she had a last-minute scheduling change, so she asked me to take over. My name is Ashley Point, and I am the president of the Koolen-de Syndrome Foundation. Before we get started, I just want to say thank you to all of our community, our patients, our families, our researchers, our donors, everybody who supports us, because we wouldn't be able to do all of these webinars without you. And with that, I will introduce everyone to James Schmidt. He is with the University of California, San Francisco. He is a research assistant within the Dr. Helen Willsey lab. The Willsey lab is located within the new Whale. Am I saying that right, James? Well Institute.
Yeah.
Well Institute for Neuroscience, which offers some of the most cutting-edge resources and instrumentation to study the cellular process involved in neurodevelopment. James and Dr. Willsey have been devoting a significant amount of their time towards understanding the role of cancel-1 in cilia formation and function. And they've been doing this in several ways, which includes generating a KdVS frog. So we have little KdVS frogs hopping around the world, which are really cool. And I don't know if he'll get a chance to get into that today, but maybe it's very cool how they can divide the frog, and half can be a typical frog and the other half can be a KdVS frog. It's very interesting to me. And then James was also talking about his role in collecting that data through nitrous oxide measuring and some other fun things that they found. So James, hopefully I introduced you somewhat, and I will let you take it away.
Yeah, that was awesome. Well, thank you very much. I'm super excited. And I feel like we'll just jump in.
Sure, go ahead.
Should I share my screen? I have a
Yeah,
little
if you have a presentation to give, that would be great. I will turn myself off, so it'll be all you, and then I'll just be here ready to answer any questions.
Cool. Wonderful. So I think my slides are shared now, so you should be able to see it.
They look great.
Awesome. So cool. Yeah, I'll get into it. So I just want to preface, and Ashley kind of mentioned it a little bit, but the families and the community of the KdVS Foundation have been absolutely wonderful and incredibly helpful for our research here at the Willsey Lab and just everything we've been doing. So I just want to preface that with a thank you. And it's been really exciting to be a part of this, and I'm really excited to share this research. So with that, I'm going to talk about cancel-1 and cilia today. So before I get into the nitty-gritty and the science, I want to give a quick background and just a roadmap of what we're going to be talking about today. I'm going to do a quick background of cancel-1. I'm sure that everybody has heard it, but I just want to give a quick summary of its known functions. And then I'm going to talk about the new function that we've discovered here at the Willsey Lab. And then I'm going to talk about some patient survey data that I've been in collaboration with Anna on, and she's been incredibly helpful with. And then talk about our frogs, and then some real-life patient data with the nasal nitric oxide that has been really exciting. So cancel-1 has before we go to cancel-1, there's two major cellular structures that I'm going to talk about today. There's DNA that we all know, and it's in every cell. It is our genetic material. It makes up everything of who we are. There's also something called tubulin. And tubulin are incredibly important for these structures called microtubules. And they provide the cell rigidity, help organization, the cytoskeleton. They are incredibly important for communication. There's just a vast amount of roles that tubulin have in our cells. So those are the two structures that I'm really going to talk about today. And we know that cancel-1 works on the DNA. It regulates acetylation of DNA. And that allows some DNA segments to be read to then create protein at a higher level, lower level, whatever it needs to be done for the cell. And this picture here on the right, you can see that cancel-1 in the big green area is in the nucleus, and that's where the DNA is. Cancel-1 also works outside of the nucleus on tubulin. So there is a paper that came out in 2015 that established tubulin's role on microtubules in cell division. And in DNA, cancel-1 regulates acetylation, and it's doing the same thing here on microtubules. So we're starting to see this emerging role of cancel-1 outside of the nucleus and having this dual function. And the main question that this is arising for us is cilia are made of tubulin, and cilia are what we study here at the Willsey lab. And we're starting to think, hey, if cancel-1 interacts with tubulin and cilia are made of tubulin, what does cancel-1 do? Or is it on cilia? Is it on the microtubules in cilia? Is it not there? We don't know. And well, we know now, but that was our preliminary question in our guiding research. So before we answer that question, I want to talk about cilia with you guys real fast. So there's two types of ciliated cells. There's primary ciliated cells and there's multiciliated cells. So a primary ciliated cell, I think the best way to think about it is if you make a fist and you stick out a finger, the cell is your fist and the cilia is your finger, and it's out in the environment. It is receiving signals, it's sensing the area. Neurons have a primary cilia, so it's in your brain, but also every single cell in your body has a cilia, except for red blood cells. They're their own special, unique type of cell. But these cilia are incredibly important and they're present. There's this other type of ciliated cells called multiciliated cells. The metaphor that I like to think about it for these cells is when you take your car into the car wash and you have the big spinning cleaning things, those are like a multiciliated cell. And we have those in our brain, specifically in the ventricles, moving fluid, but we also have them in our lungs and other areas in our body too. So there's these two types of cells. They both have cilia, some only one cilia and some a lot of cilia. Now, individuals that have dysfunction in their cilia have something called a ciliopathy. And there's a vast amount of phenotypes that come with ciliopathies. But the two things that I really want to focus on are that cilia defects alone can cause intractable epilepsy and intellectual disability, which I think are two phenotypes that you have all heard or experienced. But also, through our research and being introduced to KdVS, we've started to informally see that there's a lot of overlap with ciliopathies. So I put a star next to everything that I've come across while reading, interacting with families, and seeing that there's a lot of overlap. And that's guided some more research and really established that we want to look at this formally to see if there is a relationship between these two. So how we're going to do that is first, we're going to document the prevalence. We want to formally investigate, is there an overlap between ciliopathies and KdVS patients? We next want to look at our model system to see if we can manipulate and see phenotypes. And the model system that I think you've all heard of as of now are our frogs. And then we're also going to look at some real-time patient data with nasal nitric oxide that I will get into earlier. And some of you may even have met me and done the study. But that's how we're going to go about answering this question. So in collaboration with Anna and Anya Turala, we got this huge data set. And again, just incredible thank you to them for giving us that data set, but also an incredible thank you to all of the family members and families that filled it out. It was a long survey, very in-depth about different health questionnaires, background information, and then I was given the benefit of looking at it and being able to read it. And what we got is we had 99 families fill it out. And on the left is the bar graph showing the percent prevalence of different types of phenotypes. And the ciliopathy big three are heart defects, respiratory issues, and skeletal defects. And you can see that it's about 50% for heart, 60% for respiratory, and almost 80% of KdVS patients have skeletal defects. So there's a real clear overlap between the two. But when we go into respiratory issues and heart defects, what I find interesting is with respiratory, there's a high rate of croup bronchitis and pneumonia. That could be from or mucus clearing from maybe cilia problems in the lungs. And then also in heart defects, there's a lot of structural abnormalities. And that's incredibly important as the structural abnormalities maybe could be arising from the cilia communication and not being able to fully interact with their environment as they should. So we have this formal, clear cut, there's overlap with ciliopathies. So now we're going to investigate it in our frog model. So before I get into that, I really want to just briefly talk about why we use frogs. And there's a lot of reasons, but one of the first reasons is they have a diploid genome. And what that means is you have half your DNA from your biological father and half your DNA from your biological mother. So that is incredibly important as it's very similar to us humans. They also lay about 4,000 embryos a day, which is a lot. And with those embryos, we can do a lot. We can do genetic manipulations with injecting the embryo. So you see on the right, I have a figure that if we inject half the embryo at the tadpole stage, only half has the mutant. So what we can do with that is we have an intramolecular and also intra-organism control, which is really, really nice. And also with the rapid development, in a few days, we can have an embryo go to a tadpole and we can put it on a microscope and look at it immediately and see results. So it's really exciting. We have a large volume and it's really fast. So up to this point, we've set the stage of cancel 1 being in the nucleus, working on DNA, and then extending that to outside of the nucleus being on microtubules. And we know that microtubules make up cilia, and KdVS patients have an overlap with ciliopathies. So to investigate that further, we're using frogs. And the best way of using frogs in this manner is their epidermis. So the epidermis is the skin. And as you can see here, I talked about it a little earlier, but those look like multiciliated cells. And these multiciliated cells are right on the external skin surface. So what we can do is we can inject a manipulation or stain the frog tadpole and immediately put it on a cover slip under a microscope and look at the cilia right there. These cells are really big, so they're super easy to see. And they're also super easy to understand if there's any phenotypes. Are they smaller? Are they bigger? Are the cilia abnormal? Is their length long and short? It's really easy to immediately see how they are. And it also is giving us the foundational knowledge to see how proteins interact with these cilia. So initially, to make sure that cancel 1 is on the cilia, we did some antibody staining. So on the left is a merged image, but the pink are the cilia. We use acetylated tubulin to mark cilia. And the green is the cancel 1 protein. So you can see that there's a much higher concentration of green on the multiciliated cells. And I think the best picture to look at is the one that has the 5 picometers. And you can see that there's green dots along the axonemes, along the cilia. So there's individual cancel 1 proteins directly on the cilia. And then my image on the far right is the human cancel 1 version. So what this is we inject a human cancel 1 plasmid connected to GFP. So the frog sees the human DNA and it's the same. So it reads it, it translates it, makes it into a protein. And then wherever the human cancel 1 protein should go, it goes in the frog. And we can see that it is also on the cilia. And this is important for twofold. It shows that the human cancel 1 and the frog cancel 1 have similar localization. And that's really exciting because it then shows that maybe they have the same roles, which allows us to draw connections between frogs and humans, as we're pretty different organisms, as you might know. So we now say, all right, cancel 1 is on cilia. What happens if we deplete it or we decrease it? So with that, we have a phenotype. So in this situation, again, we took the embryo and we injected a depletion. And the control on the left, just making sure that the injections weren't causing any issues. And the cancel 1 depletion on the right, you can see a very stark difference. The cell is smaller, the cilia are clumped, they are shorter, they don't seem free-flowing as the cilia should. And you can visually see a difference, but we also statistically measured it too. So I measured the pink intensity, the tubulin intensity, to show how much cilia was there. I measured the cell surface by going on a computer program and tracing the cell size. And we also measured polyglutamylation, which is another way to see how stable cilia are. And I did this across about 8 different embryos on about 50 cells. And we saw a really clear change in structural phenotypes from the control to the cancel 1 knockdown. So we have this phenotype, but then what we wanted to say is, can we rescue it? Can we inject the human cancel 1 version into the frogs along with the depletion and see if we get a return back to baseline? And we did, but not fully. We got a partial rescue, which is still really, really exciting. So again, you can see the control. You have a big cell. The cilia are free-flowing, and the polyglutamylation in this is actually in the yellow. So you can see that as well. And then on the cancel 1 depletion, you see that the cell is smaller. The cilia are a little bit more clumped. They just look different. And then on the bottom row is the depletion with the co-injection of the human cancel 1. And here, the cell size returns a little bit to normal. Their cilia look a little bit more normal as well. And then the green is the human cancel 1 attached to GFP. So we could make sure that that was in the cell. And then again, I went through and did all the statistical measures, and we did see statistical significance with the acetylation, not so much the polyglutamylation, but my data looks a little interesting there. It doesn't have a normal distribution, so that's something I want to repeat. And then also on the apical area, we have some statistical significance. So this again is also really exciting and brings us to our next steps that I'll talk about in a little bit. But we've now gotten to the point where, again, we have the role of cancel 1 in the nucleus, and then outside of the nucleus on tubulin. And then we established that it's outside the nucleus in frogs, and then we've gotten rid of it. We saw a phenotype, and then we were able to partially rescue that phenotype as well. So now I'm going to talk about my favorite part of this project and the times where I get to interact with patients and meet the families and hang out with the kids, and it's just a joy. But that is our nasal nitric oxide study. So nasal nitric oxide is a way to measure cilia functionality within our lungs. And what happens is cilia, the multiciliated cells that line your nasal cavities and your lungs, passively produce nitric oxide. And nitric oxide is important for twofold. It helps keep cilia beating at a normal frequency, and it also helps kill bacteria, which is helpful to make our lungs healthy. So the NIOX machine takes a small tube and places it right on the outside of your nostril and measures the amount of nitric oxide for 30 seconds. It's super quick, it's super easy, and it gives us a value that can tell us how cilia are functioning. One of these things that's been interesting is the NIOX machine has been used to diagnose and investigate primary ciliary dyskinesia. So individuals with ciliopathies have incredibly lower values for nasal nitric oxide. So our lab is asking the question of, all right, ciliopathies have low nasal nitric oxide. We're starting to see cilia overlap with KdVS and other rare genetic disorders that we study. Can we use this NIOX machine to measure cilia function in patients? And with that, I've gotten 5 patients, which isn't a lot, but even with 5, we have statistical significance in the Cancel 1 population, which is really, really exciting. What we did here is we paired each test with an unaffected family member. And that's super important as it controls for other genetic factors that might influence nasal nitric oxide. So we either tested a sibling or a parent and used that data as a control value. And it was really exciting to see this, and something that I'm really looking forward to is testing more patients. So I will plug it now and I will plug it again probably in like 2 minutes. But if you're ever in San Francisco, if you ever get an email from Ashley or Anna saying that I will be in your neighborhood, I would love to meet you. I would love to test your children, you, anybody. It would be awesome. But that brings us to where we're at and what we found. So we found that Cancel 1 is on the cilia. It's on the cilia of frogs, but the human Cancel 1 version is also on the cilia. We then depleted Cancel 1 and got a ciliogenesis phenotype. We got smaller cells. We got cilia that look abnormal. Then when we reintroduced the human Cancel 1 version, we were able to partially rescue. And then we were able to see that KdVS patients have lower nasal nitric oxide levels. So what we're going to do next with this information and data is I want to increase the rescue to see if it is dose dependent. I want to see if the partial rescue can increase to a full rescue or increase the partial rescue a little bit. We also want to do different depletion methods. So we want to use CRISPR, we want to use morpholino, we want to validate these, and we also want to see if the same phenotype arises from different types of depletions. And then one thing that is really important to investigate is overexpression. So I talked about overexpression in a rescue from a depletion, but we also just want to overexpress on our own. And that's really important as we start talking about therapeutics and drug testing in the lab to see how if we can change cilia activity. Because if we increase cancel 1 too much, does that cause a problem? When we start doing drug tests in the lab, is there an issue if we do too much? And some of the drugs that we have some ideas about are Ravicti and butyrate. So one thing with frogs, it's really easy to do a drug test as we can inject the depletion and then put them in a test tube or a dish and just drip a little bit of the drug in the water and they absorb it. And then we can see how it interacts with the cilia and if it causes any rescue or if it increases the phenotype. All that we'll investigate soon. And then again, we really want to increase our sample size for nasal nitric oxide. If we only have 5 patients and it's statistically significant, there's just a whole world of where we could go with more people and more individuals. So we're really, really excited for that. And that brings me to the end. And I want to say thank you to a lot of people, but I really want to say thank you to the KdVS families for doing the participation in my NIOX, doing the participation in the survey, being so welcoming in your community. It's just been a joy to be a part of this in researching Cancel One. A huge, huge thank you to Anya Tarala and Anna Falzer for letting me use their data. You guys put an incredible amount of work into creating the survey, reaching out to families. It was wonderful. And yeah, that's everything I have for you guys.
Thank you, James. That was very interesting. I mean, I love all of that, the frog and the research and the nitrous oxide. That to me is very interesting. I did just ask if anybody has any questions, feel free to put them in the chat and we can ask James all of our very probing questions. But it's funny, you answered one of my questions, I think already. My question was going to be, why do you think the partial rescue? Why do you think not a full rescue? And I know you said maybe upping the dose would be helpful.
Yeah.
Any other ideas kind of to work with that?
Yeah, the 2 ideas we have for increasing the rescue is again increasing the dose.
Mhm.
And then uh also increasing the dose of the human cancel one, maybe
Mhm.
in coordination with the 2 drugs that we're talking about, butyrate and Ravicti. We know that Ravicti is in a clinical trial right now for epilepsy, and butyrate is already just over-the-counter supplement. So it's really, they're accessible, they're easy for us to use,
Mhm.
and we just ordered a few of them yesterday. So we're excited to start using that soon to see if that helps with the rescue.
That's awesome. And also in your thing, you talked about cilia beating. And so cilia beats in just the lungs or throughout the whole body?
So the multiciliated cells, those beat throughout the body. So in the ventricles of your brain,
Mhm.
there's these fluid areas,
Mhm.
and it's really important for that to be moving. And uh, multiciliated cells do that. And also there's multiciliated cells in other organs that also beat to help move fluid, and also just sense the environment as well.
And so you're finding, does the nitrous oxide testing show that people with KdVS and obviously the other syndromes and diseases you're studying, they have dysfunction of that beating, or is it just they don't have enough cilia?
So the answer to that question is unknown. The only thing that we can come from that is they have lower nasal nitric oxide levels, and that is related to uh, dysfunctional cilia. What that dysfunction is, we don't know yet. Um, and it could be different from the dysfunction we see in the frogs, but there is a correlation of dysfunction, which is really exciting.
Oh, it's all of it's very exciting, I think. And I think it's interesting how I know you found other syndromes that have dysfunction as well. It's not just
Mhm.
KDVS, there's actually a lot of, can you expand upon that?
Yeah, so there's a paper coming out very soon with SYNGAP,
Mhm.
and we found a correlation there. And that was awesome because I went to a patient conference and I tested 25 people, 25 families. So I got a large sample size and it was really clear that there was a decrease. So an increased sample size, seeing more patients is always really helpful.
Yes. And we hope
And
to have you at our summit in 2025 in Chicago. If we don't see you before that at another gathering, we're like, oh, we've got to get James out there
yes,
because it would have just, I know we tried so hard to get you to our July meeting, but it just with the paperwork and all that, it didn't work out, I think.
yeah, it
Didn't
was
have approval
too,
yet for that testing, but it came like a month later. And we're like, oh, it would have been great. We had, 75 families just all there ready to test. But
yeah,
we'll get you out there for sure. And I know that's a great plug. If there are any families in the San Francisco area, or we have a few meetings, I know, James, you said or Anna told me that you are probably going to go to some other conferences in 2024. And so once
yep,
we get that schedule, we can, find families in the area, and then other families can, reach out and say, actually, I'm going to be there and I want to get tested. It's so nice. It's such a non-invasive testing. I mean, it's not even
yeah,
as bad as a blood draw. We've done that on a lot of our kids. And so this is
yeah,
an easy test to ask families and patients to undergo.
yeah, yeah. And I can get you in and out in 10 minutes.
I know, it's
It's
awesome.
really fast.
It's real quick and easy. It's not, it takes a little, almost as little as a picture. So that's wonderful. So, let me see, are there any other questions right now? I don't think there are, but we really appreciate you taking the time today, and just kind of let everybody know that James is, like I said, part of Dr. Willsey's lab, and Dr. Willsey is a recent recipient of a KdVS Foundation grant. And we're just excited to continue our work with James and with Dr. Willsey, and see kind of where this goes. And if we're also, in addition to being a chromatinopathy, if we're also a ciliopathy, and how that ciliopathy plays into treatments and stuff, because we know as a family is that, yes, our kids have more respiratory issues, but it's good to see it,
Mhm.
like it actually research is into this, and to see what you show and how we can maybe find treatments for that. So we're very excited for your research, and really look forward to continue our work together.
Yeah, absolutely. It's been really exciting.
Great. Well, thank you so much, James, and we'll talk to you soon.
Awesome.
Thanks.